Okay, so hi everyone, welcome to tonight's STEMinar Night guest lecture. This is our final one of the spring 2024 semester. And so for any of you who are joining us for the first time, the STEMinar Nights are a collaborative event put on by DIYbio, the Biomedical Engineering Society, and Women in Biochemistry, with the goal of hosting a variety of speakers who come from different areas of study within science and engineering for us all to be able to learn a little bit more about the possibilities of a career and research in STEM. And so I think we can start by having each of our clubs introduce ourselves briefly. So Naina, do you want to start us off?
Yeah, for sure. So, hey guys, my name is Neda, and I'm the president of the Biomedical Engineering Society, or BMES. So, we're a club meant for people interested in biomedical engineering. We have a lot of fun social events and a lot of committees that people can join. We host lab tours of BME device companies around the valley, and also have a lot of volunteering opportunities for our members to get involved in. Other than that, we also have a mentorship program and a lab shadowing program. So if you're interested in learning more about BME, then yeah, get involved.
Okay, thanks, Naina. Is anyone, I know Haley couldn't make it from Women in Biochem, but is anyone from her e-board here? If not, I can do her intro for her, I think.
I'm on next year's A-board, but um
Go ahead.
okay. Oh yeah, we're women in biochemistry. Um we'll be having meetings like next year, probably bi-weekly. We do all sorts of things like game night, or we do little presentations, or like little DIY science related stuff. So yeah, anyone can join.
Awesome. And then really quickly, DIYbio is a student org that is open to students from all majors and backgrounds who are interested in synthetic biology, biotechnology, and genetic engineering. And so to that end, we host hands-on workshops to teach you about wet lab skills. We also host guest lectures like this one, and we also put on social events with the goal of spreading SynBio knowledge to ASU students and fostering a strong community of bioengineering enthusiasts at ASU. And so with that being said, I will go ahead and introduce Dr. Brophy really quickly as well. So tonight's guest speaker is Dr. Jenn Brophy, who is currently an assistant professor of bioengineering at Stanford, whose lab focuses on developing new tools to be able to engineer plants to contribute to agricultural sustainability and mitigate the negative impacts of climate change. And so she received her PhD from MIT, working in Dr. Chris Voigt's lab, and then went on to do a postdoc in Dr. Jose Donani's lab at Stanford. And aside from her research work, she also has contributed quite significantly to outreach efforts to introduce synbio education to students of all backgrounds, which I really admire and I think is equally important part of the bioengineering space right now alongside research. And so that was just my brief introduction. I will hand it off to you, Dr. Brophy, whenever you're ready.
Awesome. Thank you so much. The presentation that I have prepared today is all research, but I'm happy to talk about outreach and outreach teaching if that's of interest to people. I completely agree. I think it's super important. I'm going to share this PowerPoint presentation, which hopefully you can see now. Yes. Okay. See a nod. Amazing. I'm really annoying, and this presentation, at least at the beginning, is interactive. So hopefully people are willing to come off mute and answer some questions that I have. I'm going to talk today about plant synthetic biology, and I want to start by motivating an interest in agriculture and in plant genetic engineering more broadly. So this is a graph that I stole from this 2008 Nature Geoscience paper, where they're looking at the growth of the human population over time. And they're looking at it in a couple of different ways. One is just total growth of the population. So can you see my cursor? Perfect. This solid black line is growth of the human population. This dashed black line is growth that's attributable to the Haber-Bosch process. Now, the Haber-Bosch process is a synthetic way of fixing nitrogen, of getting atmospheric nitrogen into a form that can be used by plants. So it's essentially the invention of nitrogen fertilizer. And you can see that a very large portion of this population growth can be attributed to the development of this nitrogen fertilizer. In fact, this red dashed line shows you what the human population might have looked like had we never invented this fertilizer. So the development of this agricultural advancement was extremely important. And it actually garnered 2 Nobel Prizes in chemistry, one for Fritz Haber, who was awarded in 1919 for the synthesis of ammonia from its elements, the other for Carl Bosch, who about 12 years later was awarded the Nobel Prize in recognition also for his work that enabled the Haber-Bosch process, or enabled the creation of this fertilizer. Now, the interactive part comes here. There's actually one more Nobel Prize that was awarded for this transition, and I'm wondering if anybody knows what it is. So this huge population growth, you know, they're attributing it to Haber-Bosch nitrogen, but there's something else that had to happen in order to enable the increase in food production that we got from chemical fertilizers. Does anyone know what it is? Or want to take a guess?
Like the genetic engineering of plants.
The genetic engineering of plants. Okay, interesting. So no, not quite. So this is pre our capacity to be able to do genetic engineering, but I am kind of misleading you in that, yeah, this whole presentation is about genetic engineering of plants. It's actually about breeding. You guys shouldn't feel bad. I just gave this presentation, or at least this part of this presentation, at a Stanford bioengineering faculty meeting, and no one knew the answer there either. So you're not alone. The 3rd Nobel Prize winner is actually Norman Borlaug. He won about 50 years later, and it's actually for the breeding of semi-dwarf wheat. So there was no genetic engineering, but through a series of breeding events, basically taking a plant, a wheat plant that was short in stature, and crossing it with a wheat plant that was high yielding and usually used in agriculture. He was able to breed these high yielding, what we call semi-dwarf varieties. And now these are important because you can apply fertilizer to them, and instead of getting really tall, these plants produce more grain, and that prevents them from lodging. They would, the wild type plants, or the original wheat varieties, would fall over in the field if you applied too much fertilizer to them. And so it's the twin inventions of this nitrogen fertilizer and semi-dwarf wheat that give us this huge capacity, or huge increase in our capacity to make food, and this capacity to feed a whole lot more people. So I like motivating it like this. There's a lot of other reasons to try to engineer the size and shape of plants, or to try to modify them. But I think this is a nice example, especially because at its core, this change in wheat is a genetic change. If we can control genetic engineering in plants, we can probably retune them to be whatever size and shape we want. Okay, so my lab does that. We try to engineer the size and shape of plants in order to make them more environmentally resilient. What we've figured out is that engineering plant form requires really precise spatial control over gene expression across the plant. Now, to illustrate this, I'm going to show you 3 different Arabidopsis plants that are growing on agar plates. The first is this wild type plant, which has a long primary root that goes down in the direction of gravity. It's got several root branches that also grow down in the direction of gravity. And if you were to zoom in on it, you would see that this entire root system is covered in tiny root hairs, which are structures that are important for water and nutrient acquisition. Now, if you make a single base pair change in this plant that actually modifies the sequence of an important developmental regulator, you can make several structural changes in the plant simultaneously. So here the plant doesn't make any lateral roots. It's also not making any root hairs. Its primary root is shorter, and it's got a hard time sensing gravity, which you can tell because it no longer grows straight down. And there are several structural changes in the shoot as well. Now, one of the reasons why this one mutation has so many different impacts on the plant is because the protein that's mutated is important for the development of several different plant structures. It's actually expressed in all of these different tissues, and the mutant form of the protein interferes with the correct development of those tissues. Now, if we instead take this same gene, this same protein, and we express it using a promoter that's only active in one cell type, we can make a targeted change in plant structure. So here the plant does not produce any lateral roots, but it's otherwise identical to the wild type plant on the left. So we have this strategy for engineering plant development, which is to control very precisely spatial patterns of gene expression in order to modify the tissues. Okay, now if you want to control spatial patterns of gene expression in a multicellular organism like a plant, you can look through the literature for promoters that have published expression patterns. Something like this on the left is an example of what you might find in the literature, where a predicted tissue-specific promoter is used to drive expression of something like YFP, like a yellow fluorescent protein, and then you image, in this case, the root tip of the plant and see where the yellow fluorescent protein is expressed. And so you can look through this library of different promoters for one that has an expression pattern that you think would be useful, and you could use that to do engineering. But you can't always find promoters in the literature that have the expression pattern that you want. And so our lab has a workaround for that, and we use logic gates in order to control spatial patterns of gene expression. So the way we do that is shown on this slide. What we would do is take two known tissue-specific promoters. So in this case, promoter A is a promoter that's on in the root cap of the plant. And you can see that with this green color, it's supposed to be a sort of fake fluorescent protein. Promoter B would be a promoter that's on in a subset of root cap cells called the columella and in the central vasculature. If you combine promoter A with promoter B using a logical AND gate, you should get expression only where promoter A and promoter B are active or in these columella cells. So just by applying a synthetic genetic circuit that does logic operations in plants, you can generate new patterns of expression. There are actually 16 different ways that you can combine two inputs. And so in theory, you could make a lot of different expression patterns from a very small number of characterized tissue-specific promoters. Okay, so when I first started in plants, I needed to have some basic building blocks for constructing synthetic genetic circuits. We chose to build these circuits out of transcription factors, and so we made some synthetic transcription factors that are composed of bacterial DNA binding proteins fused to transcriptional activation domains and nuclear localization signals, which just bring these proteins into the nucleus. They'll bind DNA and recruit RNA polymerase to initiate the transcription of downstream genes. We also built repressor proteins that are capable of turning off gene expression. These are just the bacterial DNA binding protein fused to a nuclear localization signal. And when their binding sequence that they like to bind to is put into a promoter or a stretch of DNA that would otherwise recruit RNA polymerase to initiate the expression of downstream genes, it should interfere with the assembly of the transcriptional machinery on the DNA and prevent the expression of downstream genes. So these are the designs of synthetic transcription factors to control gene expression in plants. We also designed promoters that these things should either turn on or turn off. And you can see here like 6 binding sites essentially for the synthetic transcription factor upstream of a minimal promoter. And these are designed so that they're only on when the synthetic transcription factor binds to them. And these are full-length promoters that are constitutively active. They're always on with one binding site at the end that should shut off the promoter when the transcription factor is bound. So those are the basic tools. They're based, so the bacterial DNA binding proteins are all homologs of TET-R, which is a very famous bacterial DNA binding protein. And they have been used, they were identified by Chris Voigt's lab at MIT, and they were used to construct synthetic genetic circuits in bacteria. So we know exactly what sequences of DNA each of these binding proteins like to bind, and they made a really good starting place for the synthetic genetic circuits that we built in plants. Okay. This is one of my favorite things. When I moved from bacteria into plants, I was trying to figure out how you could measure gene expression control, and I came across these transient expression assays, and I think these are wild. The way that these work is you would take the DNA that you want to deliver to a plant cell and transform it into a special type of bacteria called Agrobacterium tumefaciens. Then you fill a syringe full of this bacterial solution and you inject it into a plant leaf by pressing the syringe. Now there's no needle here, just the syringe onto the bottom side of the leaf, and the bacteria will then fill the airspace of the leaf. And every bacteria that comes into contact with a plant cell will then deliver DNA into those plant cells through conjugation. That's trans-kingdom conjugation from bacteria into plant cells. So I thought that was wild, and also feels very crazy to like take this syringe and press it up against a leaf. But it works. And the assays that we did, we would sort of split a leaf down the middle and we would infiltrate a control construct on one side where there's just a synthetic promoter upstream of GFP and a test construct on the other where we provided the synthetic transcription factor that should turn on expression of GFP. And when these things work well, we get data that should look like this, where you get strong GFP expression where the synthetic activator is co-delivered with the synthetic promoter, and no expression when the synthetic activator is missing. In all of these experiments, we have a constitutively expressed M-cherry on the piece of DNA that's transferred into the plant cell so that we can make sure that this whole process of infiltration and trans-kingdom conjugation is working well. And that any absence of expression that we see is not the result of some sort of technical error, technical failure. Now, we can take these leaves and actually take hole punches from them and place the hole punches at the bottom of a 96-well plate and then measure gene expression in a plate reader and get data like this, where we have quantified how much M-cherry or GFP fluorescence we're getting from a leaf. Now, for the rest of this talk, we're going to be reporting this ratio between GFP and M-cherry. All right, so we tested 10 different synthetic transcriptional activators versus by 10 different synthetic promoters that they should turn on. And what we found is that there was a subset of 7 of them that were both active and specific. So these transcription factors are able to turn on the promoters that we designed them to turn on and not others in the set. That specificity or orthogonality is really important because you want to be able to use multiple of these transcription factors in the same cell, and you don't want them to be turning on promoters that you didn't design them to turn on or you didn't expect them to turn on, because that can cause your circuits to generate a fault. What we noticed is that there were some of these transcription factors that weren't quite as good at turning on gene expression as others. And so we did some really simple protein engineering to try to improve their activity. Here, we just changed the activation domain. So if you remember back on that slide, there was an activation domain fused to each DNA binding protein. The original one that we used is VP16. This is a really common synthetic or activator domain. People use it often in eukaryotic systems, all different kinds of eukaryotic systems. But VP16 is actually from human herpes virus. It's kind of amazing that it functions as an activator in plants at all. But we swapped it for an activation domain from a plant transcription factor called ERF2. And when we make that swap, we can get an almost 10-fold increase in the activity of the synthetic transcription factor, which was really nice. We were starting to get this like really simple way of tuning expression levels. The synthetic repressors that we built did not work quite as well. So here's data showing you the activity along the bottom of just these constitutive promoters with DNA binding sites introduced to them. And you can see that the constitutive promoters are all pretty strong. But when we add the repressors that are supposed to turn them off, most of the repressors don't do very much. Now, this bar is in the wrong place. It should actually be right here. So for this AMTR transcription factor, we thought, okay, maybe the mechanism of repression that we're using is not working that well, and we should try a different mechanism. In eukaryotic systems, repression is usually an active process, so just binding the DNA, kind of sterically interfering with the assembly of transcriptional machinery there is probably not working that well. So what if we fuse known plant repressive domains to it? Can we get better repression? And when we did that, we saw sort of slightly better repression, but nothing very impressive. What did help to generate much better repression was actually to move the location of the DNA binding site into the promoter. So instead of just having it at the end of our constitutive promoter, we moved it right next to the TATA box, sort of one of the important regions in a eukaryotic promoter for controlling gene expression. And when we did that, we moved from a twofold repression to 7. When we added another binding site on the other side of the TATA box, we actually increased repression even more. And then when we got greedy and added a third binding site, and we started to see the top end of the promoter come down, so it wasn't quite as strong because we had mutated it a bunch at this point. And so our fold change wasn't as good, but the repression was still quite strong. So we're excited about this capacity to be able to tune gene expression, or both activation and repression. Now, with these activator and repressor tools, we started to build logic gates. And I'm going to walk you through the design of a biological AND gate because I think it's helpful to see so that you can understand how we kind of compile these transcriptional activators and repressors to make logic. Now, in this AND gate, we're going to be controlling expression of GFP, that's our output gene. Its expression will be, or it will be downstream of a synthetic promoter that has binding sites for both activators and repressors in it. Now, there is a constitutively or constantly expressed repressor that keeps this promoter off. Now, in our 0, 0 state, where there's no inputs, then we should have no expression of GFP. In our 1 input state, our first input will be an activator that would want to turn on expression of our GFP gene. But because that repressor is there, constantly keeping this promoter off, we still have no expression of GFP. Our second input is a repressor that shuts off expression of the first repressor, but because there's no activator, the promoter is still off. So it's only when you have both the activator and the repressor that you should get expression of GFP. And we test to see whether or not these things work by the same type of transient expression assay in Nicotiana leaves that I showed you before. Here, though, we have 4 different spots infiltrated into 1 leaf, each with a different combination of input plasmid and output plasmid. What we find when we do these assays is we do get strong GFP expression only when both of our inputs are co-delivered into the plant cells at the same time, and we can quantify that too. So it's nice to see that these logic gates work. What I like about these tools is that you can actually use these synthetic transcription factors to design all 16 different 2-input logic gates. And so far, we've tested 14 of the 16 in Nicotiana leaves and found that most of them work pretty well. Obviously, being able to do logic in Nicotiana leaves is not getting us too much closer to controlling spatial patterns of gene expression in plant roots or in plants at all. And so what we started doing was using 2 different tissue-specific promoters to drive expression of our synthetic transcription factors as like the inputs to these logic gates. So the tissue-specific promoters that we used actually copy those cartoons that I showed you earlier. So here's an actual confocal image with a nuclear localized GFP. You can see that the GFP is expressed in the root tip, and that in this second one, it's expressed in that subset of root cap cells called the columella and in the central vasculature. Now, we can generate new patterns of expression by adding or subtracting these 2 together. I'm going to show you how we used one of them, one of the really simple gates, in order to start to engineer plant development. So this gate is called a buffer gate. And buffer gates are super simple. They use 1 synthetic activator. And if they work correctly, they should exactly match the pattern of a control. So on this slide, you're seeing a control promoter directly driving expression of a reporter gene. And when you use that same promoter to instead drive an activator that turns on expression of a reporter gene, you should get exactly the same pattern. So that's a functional buffer gate. And I set all this up about patterns, so you might be thinking, like, why would you do this, right? It's just a more complicated way of getting back exactly the same pattern that you had to start with. But there's a good reason for it, and that's to be able to tune gene expression level. So one of the things that we found when we were doing assays in Nicotiana leaves was that we could actually tune gene expression level using buffer gates. So on this slide, you're seeing a control construct where there's sort of a medium strength promoter driving expression of GFP. It's kind of making a medium amount of GFP. And then if you use that same promoter to drive expression of an activator, that then turns on expression of GFP with one of these 6 binding site synthetic promoters, you can actually amplify gene expression. So because you're turning on an active expression of an activator that then initiates GFP, you can boost the signal. And if you reduce the number of binding sites within the synthetic promoter, you can actually tune gene expression. So that capacity to tune gene expression is helpful because you can imagine using not a constitutive promoter, but actually a tissue-specific promoter and getting tissue-specific control over gene expression level. That's quite challenging to do usually in eukaryotic systems. We don't have the same type of control as you might in a microbial system where you could tune ribosome binding sites, for example, to preserve a promoter's activity but change expression level. So this is a nice alternative approach that works in eukaryotic systems. All right, to demonstrate that this works, we thought, well, can we tune root branching with our synthetic genetic system? The engineered plants that I showed you at the very beginning have no lateral roots, which is a nice demonstration of how important tissue specificity is for regulating plant development. But these plants with no lateral roots, they're not very useful. Plants need lateral roots to get water and nutrients from the soil. They need it to anchor themselves in the soil. If you tug on these plants when they're grown in pots, they actually just come flying right out, which isn't very helpful. And so we thought, okay, if we have this capacity to control gene expression level, maybe we can tune root branching. We already know that a large amount of expression of this developmental regulator can completely eliminate root branching. But if we reduce it, can we generate a gradient in branch density, basically tune down expression level and get more and more roots? Alternatively, do we just identify a threshold above which we completely eliminate root branching and below which we get essentially wild type levels of root branching? We didn't know when we started this project which of these things we would get, but we thought it would be interesting to find out. So we applied the buffer gates, and to be very explicit about it, this is what the constructs look like. We have a tissue-specific promoter, that one that's active just in that one root cell type, expressing our synthetic transcription factor, and then a variety of synthetic promoters of different strengths driving expression of the developmental regulator. When we did this at first, we actually found that there were no plants that made lateral roots, which was kind of annoying. Even at our lowest expression level, we were still producing too much of the developmental regulator in order to enable or allow for root branching. We could see, because we had tagged the developmental regulator with GFP, that there actually was tissue-specific tuning of gene expression level, but no change in the root phenotype. Now, one of the nice things about using these bacterial DNA binding proteins as the basis of our synthetic promoters was that the bacterial DNA binding proteins had been really well characterized. So people knew exactly what sequence of DNA these proteins liked to bind. And we had things like the sequence logo that you see here, which shows you where there are bases that are essential for the protein to bind DNA. So with this information, we were able to make a really targeted, really small library of mutant promoters that were even weaker than our 1X promoter, than the weakest one in the set I showed you before. And now with these even weaker mutant promoters, we actually were able to get this gradient in root branch density and able to show that this tuning in gene expression level was still specific. So all of the other plant features that we measured, such as primary root length, root hairs, or the ability of the plant to sense gravity, were all unchanged. So it's just this one feature of the plant that we were altering. Now, this has all been published, but I'm going to tell you some unpublished data. My lab is only about 2.5 years old, so we haven't had a ton of time to get a lot of projects started, but I'm excited to tell you about some initial directions that we're going. So first, we want to be able to use these plants that we made with this change in branch density to start to better understand relationships between form and function. Charles Darwin was really famous for figuring out that these different shaped beaks on finches had evolved over time in order to enable these birds to consume different types of foods. And a similar type of experiment could be really interesting in plants, where we start to figure out, okay, well, how many root branches do you really need for survival in different types of environments? It's really essential information to have if you want to do engineering of plants for a specific purpose, for something like better survival. You need to know what you're going after, right? You need to know what the ideal root system actually looks like. And there's a lot of theory out there about what the ideal system might be, but not a lot of ways to test those theories. Okay, so maybe we have these plants now that would enable us to test these theories about how root branch density affects survival in challenging environments. Now, in addition to just being a hard thing to have materials for, usually plant form is influenced by the environment, so it's really challenging to ask these questions. This is an example of 2 tomato plants. They're genetically identical, but if you wanted to use a tomato plant that was short, for example, to ask how low light quality affects a plant's ability, or sorry, how stem height affects a plant's ability to survive in low light environments, you would have a really hard time asking that question, because as soon as you move the plant from high light to low light, it will start to elongate its stem. So you can't really measure that correlation between stem height and light quality. But what we thought is that maybe our synthetic genetic system could sever the tie between form and the environment. So if usually the plant is responding to cues like water or nutrient availability or pathogens in the soil to change the root structure, then what if we just engineer the root structure to have a really specific shape and ignore all of these environmental cues? Then we can look really specifically at the impact that a specific root structure has on the shoot across environments. Now, there's a plant hormone called auxin that regulates plant development, and that's going to be our handle for engineering kind of environment insensitive plant development. Auxin is really interesting. It's synthesized in the shoot part of the plant and then pumped all over, including into the root system to control development. Areas where you have high concentrations of auxin will lead to, the initiation of root branches or floral structures, shoot branches, all kinds of different things. And so auxin you can really think of as this signaling molecule that's initiating expression of developmental genes. And we like it in the context of what I just told you because so many environmental signals feed through the auxin pathway. So changes in nutrients or water or pathogens, all kinds of things will influence auxin signaling. It will influence auxin biosynthesis and degradation and the rates at which it's pumped around the plant. And so what we thought is if we just eliminate this link between auxin and organ development, and then can we control organ development, right? If auxin would usually turn on expression of genes that lead to cell division or differentiation, what if we just drive expression of those things ourselves? Already, actually, this mutant regulator, this IA14 that we use to eliminate root branching, this is actually a version of an auxin response protein that ignores auxin signaling. So usually the protein would get degraded when auxin concentrations get high, and its degradation would allow for the expression of some developmental genes, but the mutant form never gets degraded, so it just ignores auxin signaling. Now we took our plants where they're ignoring auxin signaling to different degrees, essentially, and put them into different stress environments. And we saw that plants that would usually generate differences in root branching continued to generate differences in root branching across these different environments. There are features of the root system that change that we haven't engineered, for example, primary root growth. But this gradient, the fact that the gradient in branch density is maintained is really useful and is allowing us to start to ask questions about how much more root branching affects survival in these environments than less root branching. So we're excited about this, but we have been working up until this point only in Arabidopsis, and Arabidopsis is sort of like the E. coli of the plant world. It's the model plant. It's small and grows quickly, but is not an agricultural product. Nobody eats Arabidopsis. No one grows it for textiles. And so we want to try to expand our synthetic genetic circuits to other crops, to other plants. One of our first targets is sorghum bicolor. So sorghum is a bioenergy crop. Its grains, stems, and leaves can all get turned into liquid, gas, or solid biofuels. And it's a pretty high yielding plant. So at maturity, bioenergy sorghum is about 9 feet tall and generates a lot of plant biomass per unit area of land. So it's really attractive as a potential biofuel crop for those reasons. But one of the main reasons it's attractive actually is because it's pretty drought tolerant and has potential for growth on marginal lands. So on the left, you're seeing a map of the United States where marginal lands are being highlighted. And what you can see is, what I want you to know is that marginal lands are lands that are underused. They're usually difficult to cultivate, and we would not grow food crops on these lands. And so if we could grow sorghum instead as a bioenergy crop, it deconflicts sorghum or bioenergy crop growth with food crop growth, which is important for making sure we're not having negative externalities on the food system. Now, people have done analyses showing where there is potential for growing things like sorghum and switchgrass, which are bioenergy crops. And you can see that there's some overlap in areas where there's a lot of marginal lands and high bioenergy growth potential. But there's actually a lot of places where there are marginal lands and yet still very little potential to grow sorghum. It's essentially still too harsh in some of these marginal lands to grow these crops. And so we'd like to be able to improve sorghum's stress resilience in order to enable growth on additional marginal lands. But that's not easy to do. As a 9-foot tall crop, it's really hard to breed and difficult to run experiments where you can test a lot of plants across environments to see, or across environmental challenges to see how it might do. And so we've been working with the Department of Energy to try to see if our circuit tools will work in sorghum, to see if we can directly engineer aspects of the plant to improve its growth on marginal lands. Okay, so moving from Arabidopsis to sorghum is not a small task. Arabidopsis is what's called a dicotyledon plant, and it diverged from monocots like sorghum 160 to like 180 million years ago. Plants have had a long time to evolve different ways of controlling gene expression. Whereas humans and mice, for example, which I know we use mice as a model for humans, but there's many ways in which things that we discover in mice actually don't translate well to humans. And we diverged from them only about 80 million years ago. So this slide is just meant to illustrate how different these two plant varieties really are. And so to test to see whether or not some of our synthetic genetic systems work in these plants, we've been using plant protoplasts. This is sort of our equivalent to the leaf assays I was showing you earlier, but for monocotyledon species. Here, the leaves are really thin and they're really hard to infiltrate in the same way I showed you before. So instead, we take them and we cut them up into really tiny little pieces, and then we add enzymes to chew out their cell walls and can purify protoplasts, which are essentially just plant cells that no longer have cell walls or are in the context of the whole leaf. Protoplasts are really easy to transform with additional DNA, and so you can test gene expression in them. We have some initial data in Ceteria viridis, which is foxtail millet, and in sorghum, the plant I was showing you earlier, showing that we can generate these gradients in gene expression, and that the like kind of strength of the gradient depends on the synthetic transcription factor that we use. So we're excited that we can build buffer gates of varying strength in these plants, and we're starting to build logic gates in them too. Just want to highlight for you my favorite logic gate, which is called a not implies gate. Not implies gates are interesting because they're on only when one of their inputs is present in the cell. As soon as the other input is expressed, then the gate shuts back off. And what that allows you to do is refine one promoter's expression pattern relative to another. So here, if we have this root cap promoter, we can actually subtract out this columella region and get a promoter expression pattern that's refined relative to the first one. So not implies gates, when applied pretty broadly, can allow you to tune expression patterns. We can build not implies gates that work well in both Ceteria and sorghum protoplasts, and we are currently in the process of testing out all of these things in stably transformed plants. Happy to talk about what that process is like if you like. Yeah. So let me know if you're interested in that. As we move forward, we're continuing to modify sorghum. We're looking at root microbiomes, and we are continuing to analyze these structure-function relationships. I'm happy to talk more about any one of those projects, or if you want to hear about like other weirder things we're doing in the lab, happy to talk about that too. Here's the group of people that is doing this work. The sorghum and Ceteria data I showed you is by Yanina Tamborski, who is a research scientist in my lab, but a lot of people are doing a bunch of different things. So that's it. Thank you so much to you for your attention as well. I'm happy to take questions if you have them.
I can start with a question, I guess. You were just talking about how plants have like these mechanisms that they've developed over super long periods of time that they've kind of evolved to rely on. And so I guess this is something that I've come across too while doing plant synthetic biology research and reading about it. But how do you kind of ensure that what you're trying to get to the plant to do will last for long periods of time and that it won't just get mutated out?
Yeah, that's actually a problem across synthetic biology. It's even more pronounced in faster growing organisms like microbes, which will continually try to evolve away the function that you put into them. For plants, there's a couple of things that you can do for like to prevent silencing early on. Okay, so like in the first generation of your transformed plants, you can already get them trying to shut off your synthetic components. And one way to avoid that is to avoid repeated use of promoters and terminators, some of these gene expression control elements, because their plants are more likely to recognize them as foreign and then start to try to shut them off. Stability over many, many generations is tough. It's unclear exactly what design rules you might need in order to get something to really be stable in time. You can probably borrow from microbial synthetic biology to learn a little bit about how you could approach that. Obviously, like trying to make sure that the things you've introduced to the plant make it more fit can be helpful in trying to reduce pressure to evolve away from it. But some of the efforts where people are entangling sort of essential genes with their synthetic systems may end up working in plants as well. I think because, especially in agriculture, some of the plants are reseeded every year. It's not a huge deal, and it's unlikely that you would get something that's destabilizing within one generation across the entire population. So it's probably not as big a deal in this case. Ooh,
I have a question if that's okay.
please.
So you were talking about how you're using the VP16 transcriptional activation domain a lot, and then you like tried using ERF2, and also I saw on one of the slides there was another one mentioned that I think was called DREB, I think. So you're basically using these as a way to create many libraries of almost of activators, and the idea is like depending on the different activation domain that you can use this as a method to make them either stronger or weaker.
Yeah, it was sort of twofold. So in Arabidopsis, it was the change from VP16 to ERF2 was really just to see if there was a simple way of improving some of the transcription factors that were not working as well as the others. In Soteria and sorghum, so here, the ERF2 actually didn't work that well. And so that led us to try the VP16 version, which did work better. And then we thought, okay, well, maybe if we start to source activation domains that are actually from monocots, it will work better again. And so we've just sort of been exploring kind of these design spaces, trying to figure out how to think about designing transcription factors that would work in these various plant systems. But that's astute.
I also have another question. So regarding like these systems, so I don't know if I'm misinterpreting, but it seems like a lot of the engineered like transcriptional activators that you use, um, like depending on what bacteria or organism you derive the actual binding protein from, it just like binds to the motif. But are you also doing work with like having systems being like induced by external stimuli, not necessarily like, for example, like tetracycline or something like how the TET system might be functioning, but like with external stimuli such as light or like, for example, like I know that there are other like proteins in bacteria, like transcription factors that are even responsive to like gases and stuff like that.
Yeah, it's a great question. So, these TET homologs are reasonably well characterized in terms of their small molecule responsiveness. So there are some of them where, like tetracycline, there is a small molecule that is known to change their conformation and cause them to unbind or bind DNA. Most of them unbind in response to the small molecule. So we have played around a little bit with using some of those chemicals to try to exogenously control gene expression. We've also actually just been playing around with inducible systems that other people have developed for plants to try to control things like branching, for example, in response to whatever cue we would like. Developing an entirely new inducible system is not an area that we've gone to, but we have been kind of characterizing the ones that have been published before to shown and have shown to be useful in plants. And it's been interesting. Yeah, I guess one of the ways that I think about this is like plant growth is kind of variable as is. It's actually surprisingly challenging to get them to grow the same way, even when they are in exactly the same conditions or what you think are exactly the same conditions. And so adding more variables on top of it at the moment is like a little less appealing to us. But yeah, I mean, there are definitely inducible promoters that you can use in plants. And you can imagine doing things like what people have done in bacteria and mining plant genomes for promoters that are turned on in response to specific cues, or doing what we did in moving transcription factors over. I guess there are a number of ways you could develop inducible systems for plants.
Thank you.
Yeah.
Hello, thank you so much for your great presentation. I guess I kind of had a question about, well, I'm fascinated by the idea of these logic gates that are inside of the plants. I was wondering, like, a lot of plants actually codepend upon like soil ecosystems in order to like fully, I mean, grow. So I was wondering, like, is it possible to have like transgenic circuitry maybe Like, I don't know.
You mean like like from the plant to the microbe and back?
Yeah.
Yeah, for sure. There are a number of people working on trying to do that, with a variety of different soil microbial partners. So things ranging from soil bacteria to soil fungi to mixed communities of soil and fungi, of soil bacteria and fungi. There's so much to do in that space. So one of the things that we work on with Aditya Kunjapor's lab at the University of Delaware is biocontainment of engineered soil microbes. So there actually are no approved products for agricultural use that are bioengineered microbes. You can make small deletions in them using CRISPR or evolve them, but as soon as they have transgenes in the genome, then they're classified as transgenic, and we have yet to see approval of that kind of thing, for fear that like some of this DNA might, by horizontal gene transfer or other mechanisms, like get out into the environment and get out of control. And so with Aditya, we've been exploring how to couple the microbial growth to the plant and make it so that the microbes can't survive unless the plant is around. And in that case, we're engineering the plants to synthesize very specific non-standard amino acids that the microbes are then oxytrophic and require to grow. And so that's been really fun. We can get the plants to produce these non-standard amino acids. We've been playing around with trying to match kind of colonization patterns with expression patterns of these non-standard amino acids so that the plants are producing these compounds where the microbes want to grow. But yeah, I mean, there's a lot to do in that space. We have, I think, as a field, a lot to learn.
Thank you.
Yeah. Okay. I don't know. Maybe
I
that
was
was
going
it.
to ask just one more really quick question about
Yeah.
something that we were talking about earlier, just about like your work in like the SynBio educational outreach space as well.
Mm-hmm.
If you would like to like share a little bit about what you do and any like insights that you've gotten from doing that work and like kind of gaining a sense of kind of I guess how to improve the access to that sort of education and how to democratize it, because a lot of it is so dependent theoretically on being physically able to be present and do experimentation. So like how best do we translate something that is such like a physical space in science to something that anyone theoretically anywhere could learn?
Yeah, it's a great question, and I don't have answers to all of that. What we have been doing is teaching this Stanford's Introduction to Bioengineering class as a National Education Equity Lab class. So there's a nonprofit that we've partnered with that works with Title I high schools across the country. So these are economically disadvantaged high schools, and we basically get to teach this class to all these students virtually. So we've like pre-recorded lectures. We hire a whole teaching team. So for every high school classroom, there's a teaching fellow who is usually a Stanford alum that goes over, like runs basically office hours, goes over the kind of course material for the week with them, helps them through problem sets. And then the students complete the same problem sets that we give to the Stanford students who take this course on campus. And if they pass the class at the end, these students actually get Stanford credits, college credits, which are transferable. And so they can show up to university and have college credits already, which hopefully can contribute towards their major. And it's been really great. But by introduction to bioengineering, I teach this with Drew Endy, who is a big synthetic biologist advocate, and we've kind of narrowed what is admittedly an incredibly large space, right? Bioengineering could mean so many different things depending on who you are and what kinds of research you do. to basically mean genetic engineering. And then we go through like, okay, what are some like really basic things that you need to know in order to do genetic engineering, to think about the impact of genetic engineering on the world more broadly. And the goal by the end of the class is for students to be able to kind of understand new bioengineering research related news as it comes out, for them to be able to imagine potential bio-based solutions to our world's problems. So to kind of have enough of an understanding to like start to conceive of new ideas, and then to have the capacity to speak to others about bioengineering, to kind of like teach them. What we're not doing is teaching wet lab skills like pipetting and you know, cloning and measurement in a like hands-on kind of sense, because you're right, it is really hard to do. But we're hoping that with this kind of like intellectual framework, it enables students to then reach out to more local institutions and have an informed discussion about what it might be like to go do research there, so that they're not starting completely from scratch when they get there. And actually next year, we're going to be piloting this 2-week kind of summer follow-on to the course, where students who like really loved BioE 80 can sign up in small groups, and I am going to be shipping pipettes and like small electrophoresis chambers around the country, and then challenging students like, okay, go to the store, buy this thing, and we're going, sorry, like buy this type of produce and this type of produce, and we're going to see if we can detect transgenes, because you can actually buy, you know, genetically modified produce of different types. And we're going to talk about, you know, how to approach labs if you want to do research, and how to do DNA extractions, just like teaching some basic stuff. But the idea is that you would be able to do all of this like at home, at the kitchen table, which could be kind of cool too. So
Yeah, no, that's super cool. It's really nice that you're doing all of that. I think it's super necessary, like I was saying before, to be able to have people have exposure to that as soon as possible. And it's kind of like similar to what we do at DIYbio as well. We have like a workshop series where we teach people all those initial cloning skills. So yeah, I really appreciate your time. I think if no one else has any questions, I think that'll be all for today.
So that's the link to our class. If anybody wants to see it, there's like all the recorded lectures are there, and we actually have an LLM trained on all of the course data. So we have like a digital TA that you can ask questions to and stuff too. If you're ever bored, or you know somebody who wants a kind of intro to bioengineering, all that stuff is there. So
Super
good
cool.
luck. Yeah. Okay. Well, thank you for having me.
Thank you so much for your time. We really appreciate it. This was super, super interesting.
Awesome. Have a great night, everybody.
Yeah, you too.
Bye.
Thank you.
And thank you so much
Thank
for the
you.
invitation.
Of course.
Bye.